Again, I'm Cortney Kidd. I am from Floyd Central High School. This is actually my first time presenting with you all this year. When you all had the fall summit, I was out having a baby, so not as much familiarity with everybody else. But I took a different turn with my grant this year. I've done these ARI grants for a couple of years now. In the last few years, I've focused and put a lot of my emphasis on pulling technology into my biology classes. But then last year, after I saw the field test that my students had taken, I noticed that one of the areas that we seemed very lackluster in was their ability to problem solve. Like, they just weren't able to use the models that they were provided with. They were struggling to pull the content out. So this year, when I designed this grant, I based it off of the modeling premise that's in the Next Generation Science Standards. And what I did was I looked at my core units and I decided the places that my students were struggling the most with those concepts of modeling. And what I did was I designed my purpose of investigation around that. I wanted my students to be able to not only create models, which we tend to get. When we think models in science, a lot of times the kids think they're going to build something 3D. They think they're going to make a poster. They think they're going to use molecular models to build something. But they also have to understand that there's a lot more analysis and a lot more premise that goes with it. For example, like the mathematical concepts. And that was just something that my students weren't able to grasp. My big purpose behind this was to drive my students to actually use diagrams and replicas of things. I wanted them to be able to create those mathematical applications that they had seen on that field test, because I know that was something they were not common with. We were so connected to the old end of course assessments where it was just rote recall and definitions. I wanted them to actually look more at the application. My plan of action, like I said, first of all, I actually picked out that I wanted to work on modeling with the students. But the big thing for me was I had to figure out a way to make this more experimental for my students than just saying, hey, we're going to create models, we're going to use models. I had to figure out a way to make it more invigorating for them, because without that little bit of connection to everyday life, I tend to lose them a lot of times. And I was tending to lose them in areas like biochemistry because they didn't care what a carb looked like. They can't really visualize it. They knew it was in the things that they eat and they drink, but they didn't know what it looked like. I was losing them when we would talk about properties of water, because it's just like, okay, it's water. But they didn't really understand the properties and how those things were working. So what I did was I sat down and I designed my actual purchase around 3 different units. I designed it around biochemistry, where I looked at enzymes and the magnetic water kit. I also looked at biodiversity. That's something that I'm actually still working toward for the end of the year. I do my ecology unit last, so that's coming up. But then I also worked on some genetics and photosynthesis aspects of different units. Planning and prep. Implementation was probably the hardest part for me this year, and I am being completely open with you when I say, after being gone for 3 months and then being out for Christmas break, I have been a little hectic. I've been a little scattered this year, but coming back and trying to pull this directly in was something that I did struggle with a little bit. We have completed all of the tasks and the activities except the enzyme activity, and then, like I said, I do the biodiversity at the end of the year. And I will not hide the truth from you with enzymes. I was just out at that time. I could not work it back in when I came, but I have that kit that I can use next year with them. One of the first things that I did, and this is actually an example from my AP class, one of the things that I focus on in AP biology is this idea or this premise of applying concepts to look at an emergent property, how you can take small scale things and actually develop something to a larger scale and we use this to talk about like the endosymbiont hypothesis and how we have these separate organelles that come together to make a whole. What I actually did with them was we used a station activity, and I used a small amount of my money to fund like some of the properties. I bought some puzzles and things like that, where we looked at emergent properties, taking individual things from separate stations, bringing them together to be able to describe a whole. And then for reductionism, where I actually would give them something and they would have to break it down and be able to tell me how something would work. And I know at first that sounds like a really oh, well, that's probably a boring activity, but the problem was not in the activity itself. The problem was in the way that my students were communicating. If anybody in here is in an AP class or you've taught an AP class, you know that the way those students have to address the material is different than any other test that you see. And for my students, this was one way for me to develop a way for them to actually actually deliver concepts assuming that I knew nothing. They were bad to use pronouns. It did this, that did that. And this helped me to eliminate that because I made them be very picky and detail-oriented. And it was just a roundabout way for me to introduce a new modeling concept. Highly recommend these. Probably one of my favorite things that I've used since I started teaching biology. I found a company called 3D Molecular, and I actually found them probably about 6 or 7 years ago through doing Science Olympiad, and I used them for my protein modeling kits, but I actually stumbled across their 3D water kits a couple of years ago, and I only had 1 or 2. Part of my grant helped me fund buying a class set of these, and what they are is they're tiny water molecules, but they're magnetized, and it allows me to let my students model like polarity of water, adhesion, cohesion, so they can actually see the repulsion forces and the attractive forces that they can have with water molecules and other molecules of their same nature. This is the enzyme lab that we didn't get to, but it's enzyme catalysis, so they can actually look at different substrates and how they work within an enzyme-based system. We did cell efficiency. Again, this is something, and again, pulling from that test that I saw last year, cell efficiency was something that my kids understood. They could give me verbatim sentences about. They understood surface area to volume ratios, but they couldn't really pinpoint a visual aspect. What this is an auger-based kit, and I had the students do it prediction-based, where they had to tell me, which cells are more efficient, smaller ones, larger ones, and then we actually did a chemical dip dye to where they saw what could be taken in by the cells, so they would actually see why it is more beneficial for them to be smaller. We did allele frequency in 2 different ways. I bought some Indian corn, and we did a Wisconsin fast plant. I did both because sometimes just counting the corn, I think they would get kind of bored sitting there counting little kernels. But with the Wisconsin fast plants, you can actually plate those. They'll grow within a couple of days. So the kids actually were able to create their own real-life models to see how the plants actually bred and what traits they were getting within a real-life system. It's a pretty quick one to actually get through. We use this for chi-square calculations to give them a mathematical model. This is more common for my AP students than my general bio students, but I can use the corn in both, and it works out pretty well. Conclusions and outcomes. The big thing for me was going back to my TCTs and the models that we're supposed to use in classes now. I do see increases when I use modeling with my students, just for the simple fact that just recently we did a TCT, and if any of you are familiar with that bank, there's one on there where it's having them predict if a virus is living or not. And it is all diagram-based, it's model-based. They give them a table of information, they give them a graphic, and they have to interpret it. Because my students had some familiarity with those types of things, I do think it made them more confident in saying, here's what I saw, so this is my result. It's kind of like by using these modeling strategies, I'm trying to pull in a what I see plus a what it means kind of thing, because they can always tell you what they see, but they can't always tell you what it really means. And that's where I was trying to hit with this grant. Students did work well in the partner sets with these activities. I will say, for further investigation purposes, I think I would cut back on my scaling on my groups to 3 kids instead of 4. But I have high populations in a lot of my classes, and that wasn't really possible this year. But if you do have a smaller class set, that is something I would recommend. And again, I mean, everything worked out pretty good. Did have some timing issues because of the baby, but other than that, any questions? Okay. Thank you all.